Aloha, everybody. It is time to start our discussion covering the last chapter of this class, or regulation of gene expression. This is perhaps the most complicated and least understood processes related to genetic information or in molecular biology in general. It is very complex, it is very general, and during this discussion, I'm only going to cover some basic foundational principles related to regulation of gene expression. To begin, it is noteworthy to say that bacteria has been estimated to have about, in general, on average, about 4,000 genes, while humans probably have about 20 to 23,000 genes. Now, the fact that we don't know how many genes do we even have says it how well do we currently understand our own genomics. Despite that, we have a very good start, or we've made quite a bit of advancements in terms of our understanding of gene regulation. And every day we move forward in our understanding. Okay, well, to begin our discussion, let's talk about these first two questions. Think of few reasons why should there be regulation of gene expression, and think of a few examples demonstrating that there indeed is gene regulation. Well, you can take a moment and think for yourselves. But if I was asked these questions, I would say we, or all advanced multicellular organisms that are made out of various different types of cells, tissues, and organs, obviously represent a unity of specialized functions. Each organ, each tissue is designed to do certain functions. However, all these organs and tissues have been generated based on the same genome that exists in all different cells. It's the same genome. That means that there is a differential expression in different cells. So something had to make certain cells to go one route and other cells to go a different route. Something told certain cells to become skin cells, neurons, myocytes, intestinal tissue, to generate the mucosal tissue of the stomach, for example, or to generate the endothelial tissue of the vessels or the heart. That itself demonstrates that there is programmed gene regulation or differential gene expression. And there's much more to be said, but I just want you to think and realize that for a bacteria that has a single cell, that is made out of a single cell, all it needs to do is just respond to its environment and to adapt to its environment. And we as multicellular organisms also have to do that. But because we're so complex, we have to have differential expression in our different cell types. I also want to give you this example, which comes from my own research while I was a graduate student. In this research, we wanted to study the effect of this gene known as fosdeucin-like protein, or FLIP. And one way to study the function of a gene is by knocking it out completely, or by knocking it down, or knocking its expression down, or silencing it a little bit. And here in this case, we use two different small interfering RNAs. We'll talk about them during our next class. that lead to a differential expression of this gene in the cells. So we took cultured cells, we transfected, or we put in these cells these small interfering RNAs, and they led to a different degrees of silencing of the fususin-like protein expression. And you can see the expression of fususin-like protein, where wherever we use these two, we have decreased expression compared to the control cells. In addition to that, we also have a control called lamin AC, just to show that the presence of siRNAs in the cell do not interfere with our goal, which is to determine what is the expression of G protein beta 1 subunit. And we see that when the expression of fususin-like proteins falls below 80%, we see a decrease of about 40% of G beta 1 expression. Now, this is what the figure shows. This is what our results show. The question is, or the question we started discussing once we saw these observations or these results is, what are the reasons for G beta 1 decreased expression? Think about that. What could be the reasons, or what could be the reasons for G beta 1 to decrease its expression when the expression of fususin-like protein is decreased to a minimum? Well, normally I would ask you to consider that, to discuss it between yourselves, but since this is a remotely recorded lecture, I'm just going to go with it. There are really 7 general areas we needed to explore. First is, was there a problem with mRNA synthesis, or was there a problem with the transcription of G beta 1? And how did fususin-like protein led to decreased transcription of G beta 1? How do we prove that? Or was there a problem with the mRNA processing, not so much with the generation or synthesis of mRNA, but how it's processed? Or was there a problem with its stability? Or was there a problem with protein expression or translation? Or perhaps translation was fine, but post-translational modifications of the G beta 1 was somehow interfered with, and that's why we have a decreased protein in the cells. These bands, by the way, represent the expressed amount of protein. Or perhaps there was a problem with the protein transport. G beta 1 is often found, it's found on the membrane. It does get released from the membrane, but at some point it is attached to the membrane. Perhaps there is a problem with the localization of G beta 1 to the membrane. Or perhaps there was a problem with the protein stability or degradation. These were the 7 areas we had to eliminate or study in order to find out where was the problem, or what was the reason for why is fususin-like protein necessary for the proper expression of G beta 1? Well, after checking all these different areas, we determined that the reason was with 4 and 5, protein expression and post-translational modification and assembly of the G protein heterodimer between G beta 1 and G gamma 1. Normally, I would also ask you to think, and you should think, how would you normally study these areas of gene regulation? How would you study transcription? How would you determine mRNA stability? How would you determine protein transport or protein stability and degradation? This is what researchers deal with on a daily basis. Researchers that deal with regulation of gene expression, they all study one of these major areas, and there's also sublevels of each one of those major areas. Okay, well, to move on, everything that I've said in that example that I that I've just demonstrated is basically described in this figure, showing the different levels of gene regulation: transcription, mRNA, processing instability, translation, post-translational processing and protein degradation, and localization, etc., etc. All right, to move on, what factors do you think regulate transcription? Well, I've listed a few very good areas that can be regulated in order for the cell to regulate transcription. Perhaps we have different DNA availability. How well DNA is packaged, or whether it's relaxed or supercoiled, determines whether a certain area of that DNA will be expressed. Also, the promoter sequence matter. How well does the promoter sequence matches, so how well does it bind to the factors or RNA polymerase? Also, specificity factors or repressors or activators, all of these regulate, and they are important for transcription, hence for regulating transcription. A few questions we can ask in regards with these factors are: what is the role of the consensus sequence in the promoter, and role of mutations? What if the promoter mutates? Well, the promoter sequence determines the binding affinity of transcription factors or specificity factors for bacteria, such as the sigma factor or the TATA binding proteins. Mutations in the sequence of the promoter that move the binding site away from the consensus sequence lowers the binding affinity of the transcription factor, or in other words, decreases transcription that is ran by that promoter. Mutations in the promoter that improve the binding of the transcription factors or the specificity factor to the promoter will increase transcription. So you can see the role of variability between promoter and promoter. Now, what do we call the DNA sequence bound by the repressor, or where is it localized? First, what is a repressor? A repressor is a protein or compounds that bind mRNA to downregulate transcription. The sequence bound by a repressor is called an operator. It is located near the promoter in bacteria or much farther away in eukaryotic cells. Now, I know I'm going to introduce a lot of new terms here, but please take it slow and don't rush it, and make sure you really understand the meaning of these terms. Again, the repressor is the substance that does the downregulation. The sequence that is bound by the repressor is known as operator. Okay, now the next question: what is the difference between effectors and enhancers? How do they affect gene expression? Now, enhancers are DNA sequences which are bound by transcription activators. So we have a repressor that downregulates, activator that upregulates transcription. The repressor binds operators, which is the DNA sequence, and the activators bind the DNA sequence that is known as enhancers. So again, repressors bind operators, and activators bind enhancers. Okay, so how do they affect gene expression? Well, obviously, enhancers or activators activate gene expression. Now, the effector molecules are small molecules or proteins which bind and regulate the binding of the repressor to the operator, or an activator to an enhancer. In other words, the effector molecules are molecules that control the binding of the regulators of transcription. The effector molecules regulate the binding of the repressor to the operator and the activator to the enhancer. Okay, I'm pretty much out of time, so I'm going to stop this recording at this point, and I will continue in a moment. Aloha. This is the second portion or the second part of the gene regulation lecture. So last time we ended with these questions, defining what is a repressor, operator, what are repressors, operators, activators, and enhancers, and effector molecules. And again, just very quickly, repressors bind operators, activators bind enhancers, effector molecules bind repressors and activators and control their binding to their corresponding sequences, either an operator or an enhancer. All right, in this figure, we'll show two different types of negative regulation by an effector molecule. In this case, that effector molecule is known as a signal molecule, and two examples of positive regulation. In this particular case, we see a repressor bound to an operator, and there is no transcription. When a signal molecule or an effector molecule binds the repressor loses its affinity for the operator, falls off the mRNA, and then falls off, and then falls off from DNA, and mRNA is being synthesized, or transcription has been activated. In this example, we have a signal molecule or effector molecule bound to the repressor, and it's bound to the operator. There is no transcription. But when the effector molecule is removed, then the repressor loses its affinity for the operator and falls off, which stimulates transcription. So these are examples of negative regulation. Positive regulation, pretty much the same thing, but in the opposite way. We have an activator bound to an enhancer, and we have activation of transcription. We have mRNA being synthesized. When an effector molecule binds that activator, the activator loses its affinity for the enhancer and falls off. That stops transcription. Or the opposite, we have an effector molecule with the activator bound to the enhancer, and then we have transcription. When that effector molecule falls off, then the activator falls off. There's nothing to activate transcription. Transcription stops. So you can see that the effector molecules could do their job by multiple mechanisms. They can control the repressor's binding or the activator binding. They can make them bind stronger, or they can make them bind weaker. Whatever the effect of the signaling molecule, the effector molecule is, it can be done using one of these mechanisms. Now, the following two slides that I have here, I don't want you to get caught up into details. All I want you to see through these slides is the complexity of the regulation and the spatial orientation of the regulatory machinery. A lot of times we focus basically right here at the transcription window, but the controls are far away from them. You can see, based on how DNA is folded, the activator or enhancer sequences could be thousands of bases. I mean, right here it's shown this loop, but this loop could be thousands, tens of thousands of bases away, and not as small as you can see it here, but this could be far away. You have to consider this spatially in terms of how DNA is folded and where those sequences fall, or where they are localized with respect to the beginning of the gene or the promoter. Like I said, enhancers can be far away. They could be mediator proteins that can bring these sequences together, or repressor molecules, again, that can inhibit the formation of the transcription window. We have here in this figure factors that control histone modifications. Histones, if you remember, are part of the nucleosomes. Nucleosomes are basically these protein DNA formations that fold DNA in a very specific way and quite tightly. There are certain proteins that regulate the function of the histones, that relax the DNA and make it available so that then we can have a formation of the transcription complex, so transcription can actually occur. Like I said, these two slides that I just showed you, this one and this one, don't worry too much about understanding every little thing. I'm trying to memorize it. Just try to visualize it spatially and to understand that the regulation is quite complex. And of course, this is not so much in bacteria. This is in more advanced organisms such as humans or mammalians in general. All right, to get back to bacteria, in bacteria, actually, it's quite common to have multiple genes next to each other, and their expression is driven by one promoter and regulatory sequences that are usually around the promoter. Now, that complex of genes, promoter, and regulatory sequences in bacteria is known as an operon. That's not the case in eukaryotic cells. In eukaryotic cells, each gene has its own promoter, and the regulatory sequences could be far away from the promoter. All right, now let's take a look, more detailed look, at the lac operon. The lac operon is made out of 3 genes, the Z, Y, and A, that code for beta-galactosidase, galactoside permease, and thiogalactoside transacetylase. You don't have to know these names. I just read them. I actually don't even remember them other than beta-galactosidase. You don't have to know them. Just know that there are 3 genes. And their expression is all regulated. Well, I shouldn't say regulated, but is initiated by this promoter sequence P right here. In addition to these 3 genes, we also have a repressor gene, the I, probably standing for inhibitory function or inhibitor, and the expression of the repressor is driven by a promoter, PI. When the PI is activated, there is a transcription of the repressor gets expressed and then binds at the operator sequences O1, O2, and O3. The O1 has the highest affinity for the repressor, but they do bind with all of them. When they associate in a homotetramer, shown right here, you can see 4 identical repressors or in a complex. It's believed that they loop around the DNA and they prevent the expression of the 3 genes of the lac operon. Here we see a crystal structure of the DNA bound to the repressors, 4 of them, 4 different repressors. On this end, the deep side of the figure, we can see the repressors in their bound form with the kind of the shaded graphics, and you see with more solid colors repressor molecules that are now not bound to DNA. These repressor molecules that have been released or that release DNA, that release was triggered by IPTG. If you remember, in the 483 lab, we did use IPTG. Think about why did we use it? We used it to stimulate protein expression. Now you understand how that's done. When IPTG is added, it binds the repressor, and the repressor releases DNA. Now DNA is now available. The promoter is now available for expression to start. And if you have any genes that are located downstream from the promoter, and that is the promoter we normally use for bacterial expression, they will be expressed. So that's how IPTG works. It stops the binding of the repressor to the operator sequences, thus opening the DNA and making it available for gene expression. All right, so let's talk a little bit about the binding of these proteins to DNA and how is that facilitated. A good question would be, what kind of interactions would facilitate protein-DNA binding? Well, think about it. DNA, what type of surfaces DNA has? What type of interaction DNA will facilitate? Well, it's not far from the thought that we're going to have electrostatic interactions. DNA in general is negative. We can also have hydrogen bonding. And in rare cases, we could have also hydrophobic interactions, but in generally they will be polar and electrostatic interactions. The regulatory proteins interact mostly with the major groove forming hydrogen bonds with the bases. We see some examples of hydrogen bonds formed with the basis of the major groove. Now, which amino acid will facilitate such interactions? Well, we just said hydrogen bonding electrostatic interactions. Any amino acids that can form hydrogen bonding or electrostatic interactions with negative charges would be okay, would be part of this binding site, such as lysine, arginine, perhaps histidine, also asparagine, glutamine. We can see an example of glutamine right here, how it forms hydrogen bonding electrostatic interactions. Now, let's talk a little bit about the types of the DNA binding proteins and what type of motifs are observed in proteins that associate with DNA. We first have, well, proteins that associate with DNA can either associate only with DNA or they can associate with DNA and proteins. So we will look into two different binding motifs, DNA binding motifs and protein binding motifs. DNA binding motifs are named, well, these are a little bit of an interesting names, but there's a reason behind them. And we'll talk about them in more details just in a moment. But they're helix turn helix, zinc fingers, or homeodomain. These are the three types of DNA binding motifs: helix turn helix, zinc finger, and homeodomain. There's two types of protein binding motifs: leucine zipper and basic helix loop helix, not helix turn helix. There's a difference, and we're going to talk about that in a little bit. All right, so let's start looking into these motifs. The first one we're going to talk about it is the DNA binding motif helix turn helix. You can see it right here: helix, turn, and helix. These fit primarily into the major grooves. These are the binding motifs that are part of the LAC operator. You can see them here in gray. These are the helix, turn, helix. They're small, about 20 amino acids, and each helix has about 7 to 9 amino acids. The next motif that I'm going to show you are the zinc fingers. Zinc fingers are about 30 amino acids, and there is a zinc ion involved in their structures. The zinc ion is not involved in the binding with DNA, but coordinates the orientation of the amino acids so that they can associate with the DNA. The third DNA binding motif is the homeodomain, I'm sorry. The sequence that encodes for homeodomains is called a homeobox. The genes which contain homeoboxes are known as Hox genes. Hox genes are very hot research topics right now because, as you would guess, they express transcription factors, and transcription factors regulate expression of genes. So that's why they're actually quite hot topic right now in the research community. Again, we see helix turn helix. The only difference between the helix turn helix and the homeodomain is the size. The homeodomains are larger. Instead of being about 20 amino acids, these are about 60 amino acids. Okay, I have left just the protein domains, but I'm going to run out of time in a few seconds, so I'm going to record that in the third part of this lecture. Aloha, welcome back to our third and final part of the gene regulation lecture. We ended our discussion, or I ended my presentation with this slide last time. Again, let me just briefly, so we can connect well with what I'm about to say, go over the two different types of binding motifs. We have, well, the DNA binding proteins in generally have the ability to bind either only with DNA or with DNA and proteins. They facilitate this through, or it's been observed that many of the DNA binding proteins have similar binding properties or binding surfaces. There have been 3 different DNA binding motifs that have been observed: helix, turn helix, zinc finger, or homeodomain. And there have been 2 major types of protein binding motifs that facilitate protein-protein interaction between the DNA binding proteins. And these are the leucine zipper and the basic helix loop helix. Now we talked about the helix turn helix, we talked about zinc fingers, and we talked about the homeodomains. Now let's talk a little bit about the motifs that facilitate protein interaction. And a great example to begin this presentation is the leucine zipper. Now, this is a protein that has 2 different binding motifs. With one part of its structure, the leucine zipper interacts with DNA. That's this part, or the basic part. That's the part that or the DNA binding region. I shouldn't call it basic because some of you may assume that the name of it is a basic region, which is going to confuse you with the next binding motif. But leucine zippers have DNA binding region. And that DNA binding region, as you can see here from the sequence analysis, is rich on, as you can see, the highlighted ones, basic amino acids, which would associate quite well with the negative charges of the DNA. So leucine zippers have DNA binding region that associates with DNA. What makes this protein special is, well, in addition to this property, is the fact that the rest of the protein has these repeating leucine amino acids that are spanning every, 2, 4, 6, 7 amino acids. And when the 2 proteins where the dimer associates with DNA, part of the reason why the dimer associates with DNA is that these leucines from the opposite side, they associate with each other and then they hold the dimer together. They act as a zipper in a way, and they hold the 2 proteins together. That portion does not interact with DNA. That portion facilitates the interaction between the 2 protein molecules that bind with their DNA regions to the DNA and control its expression. So these are the leucine zippers. And the last example that we're going to go over in this lecture are the basic helix loop helix transcription factors, or transcription factors that contain basic helix loop helix motifs. The basic part describes the part of the protein that associates with DNA. This is the basic part. The basic part is the area of the protein that binds to DNA. The helix loop helix part of the protein is the protein binding motif. The helix loop helix motif does not associate with DNA in general. It's only the basic part of that first helix that associates with DNA. So again, the danger here is to confuse helix loop helix with helix turn helix. Remember, helix turn helix, which is right here, that motif associates with DNA. With the helix loop helix, helix loop helix, this is a protein binding motif. The basic part of the protein associates with DNA. Okay, that's pretty much the end of what I wanted to say. I hope this was clear. If you have any questions for me, please let me know. I'll be happy to answer them.